Restriction endonucleases or restriction enzymes. What in the world is that? What's a restriction enzyme? Yes.
Isn't it an enzyme that cuts like foreign pieces out to it's like a defense mechanism?
It is an enzyme that cuts DNA and it's foreign to its own genome. Okay? And we said that in bacteria, what is the biological function of a restriction enzyme in the bacteria? Why does the bacteria have them? There was a biological reason. It's immunity. Okay? And so if foreign invading bacteria gets inside that enzyme, it's going to cut it. All right, inside the bacteria, this enzyme will cut it and destroy the information. Now, restriction enzymes are not, we don't use them in the natural way in terms of, we're not going to invoke immunity for bacteria, but what we're going to do is isolate that enzyme and use it as a tool, a molecular tool. We're going to be able to cut DNA very specifically, know where the cuts are, and create cuts that are uniform. Every cut will only cut one place and all the ends will be complementary. And so the cleavage site on any restriction enzyme is a palindromic site, somewhere between 6 and 10 residues long. A palindromic site is a sequence that is read backwards and forwards the same way. So here, 5' C, 5' C, 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 G, G, C, C, G, 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 G. So it has to be palindromic. Now here is the deal. In a long strand of DNA, you're going to have palindromic sites come up randomly. Okay? And small enough sites, the shorter they are, the more often they're seen. And so they're just randomly. Now, as soon as we cut the DNA, effectively we've destroyed the information of that DNA sequence, but we're going to use these in a way in which I'm going to cut specifically a chunk out to use it. Okay? And that's in the terms of cloning and the idea that we used in terms of cloning. Now, at the time I did this slide, they say there are 90 restriction endonucleases or restriction enzymes. There are hundreds now, and it just depends upon what you need. Now, even though there are hundreds in the catalog, you don't use, or very often we don't use a large number of them. We tend to use a small handful, and you'll see these names out there like EcoR1, okay, and so, or BAM-H1. They are traditional restriction enzymes that are used to cut DNA to match things up. Now, why in the world do we use these here at Lee? Because they're cheap. And so we can buy them very inexpensively and we can use them. And most of the vectors that we are going to place our pieces of DNA have sites for these. And we actually even buy vectors that are engineered to place those sites in there. And so they're easy to use, they're abundant, they're used all the time. Of the 90, there may be 20 that are used really often in laboratories, and then you have to get strange or exotic. You can buy them, but they're not used that often. All right, these have been characterized. The genus, type, the nomenclature comes from the genus and when they were found. The restriction endonucleases, again, do not degrade the bacterium of the DNA in the bacterium that they're produced because they are modified by methylation. So that's just a piece of the natural process of restriction enzymes. The vectors that we use quite often are plasmids. That's one very often used vector, quite often used in undergraduate laboratories and in research. They're cheap. They're small circular DNAs. They are introduced into bacteria, they have these functional pieces, an origin of replication, so it will be copied. Antibiotic resistance, and we saw that because we're going to infect cells and then grow them onto a medium that has antibiotics. If no vector gets inside, that cell will be destroyed. Only cells that will grow have at least the vector in it. And then recognition sites to be cut. And so you can see EcoR1, BAMH1 are on there, the kinds of cuts that we are going to place in the sequence. Now, not all of the sites, restriction sites, are as helpful, or some are really, really helpful. And so, by chance, if I put something in the BAMH1 site, I insert DNA, what functionality does that change for this vector? If I put my piece of DNA I want to copy there at that site, do you see a consequence? What happens to that vector? It's going to lose something. It will not be tetracycline resistant. It will still be ampicillin resistant, but I lose one of those. That can be used to differentiate whether the cells are infected or not by growing them in different medium that have different antibiotics. The EcoR1 would not disrupt anything, and so you can use strategies to help you identify was DNA inserted or not, not just if it's resistant. The PBR322 vector is one that is used very, very often. You will see that in lots and lots of journal articles, and one that is used quite often in undergraduate laboratories, again, because it is cheap and we have good systems to know how to get that vector into the E. coli. We also, plasmids were actually a natural product in bacteria, and we just isolated them. One of the reasons for antibiotic resistance are these plasmids. In nature, we isolate this, and we're now using it as a tool. Some of them had natural sites where we can insert DNA, but they didn't have as many sites as we might have wanted. So what we've done is we've engineered vectors. We've taken the natural, and we've inserted a piece of DNA that has lots and lots of linkages, okay? We've placed an extra piece of DNA where I maximize the types of inserts that I can place in, all right? Also, we've added sometimes a reporter. So a piece of DNA that will make a protein, and if the protein gets, if that plasmid gets inside the cells, it will produce a reporter, or it will produce an enzyme that could be used to identify. And so here we have a plate. It is antibiotic resistant, so every one of the cells has a vector inside of it, but the white cells don't have this enzyme. And so I've inserted the DNA, okay, but that might not be what I needed. The blue in this case has an inserted plasma, but it contains the functional protein that I wanted, which means that I've actually cloned a piece of DNA and inserted. If I've got it white, then I disrupt the reporter. I have a DNA insert that I wanted, the plasma and the DNA. But if the blue is responding, that means no DNA actually was inserted. The reporter enzyme was actually made, and so the plasmid got in, but no DNA insert was there. And so you can identify all white have what you want, all blue are the ones that, yeah, plasmid got in, but no DNA was inserted. And so you can do selectivity, which is really, really easy. If you can read color between white and blue, you're okay. Even most colorblind people can do that one, of which I am, okay? Now, plasmids, you can get, there are limitations. You can only get up to about 15,000 base pairs at the maximum. Sometimes you want large amounts of DNA, sometimes you want small amounts of DNA. The larger the plasmid becomes, the less infectious it becomes. It gets harder to get inside. Transformation, or getting that vector inside the cell, is done by transformation, which is it's shocked by calcium chloride and a temperature change, and that allows some of the plasmids to get in, or you can electrify it to try to get the plasmid in, but low infection rates. To increase our infection rates, we can change the type of vector we use. Bacteriophages is a wonderful vector. Okay, it has a genome of about 50,000 base pairs. Okay, but about a third of those base pairs are really not needed for the function of the bacteriophage. Okay, and so what we can do is we can cut out the middle and plop in what we want. Now, here's the deal. The bacteriophage, when it all comes together and becomes a functional group, it's normally about 50,000. Okay? It can accommodate as small as 40,000, and it can accommodate up to 53,000. So in that range, as long as the DNA is in that range, the bacteriophage can work. Okay? So we can cut and we can package in our piece of DNA. Now, if we cut it and we package in our DNA, you get lots and lots of fragments. If it is too short, it doesn't get put into the packaging. If it's too long, it doesn't get put in the packaging. But if it's just right, just like Goldilocks, it will fit into the packaging. Now, we can pack in up to 23,000 base pairs, a little bit larger, okay? Not significantly larger than plasmids, but a little bit larger. But the bacteriophage, though, has a much higher infection rate. Bacteriophage looks kind of like a lunar lander. It will land on the E. coli, stick a tube through the cell membrane, and squirt all the DNA in. The infection rates are much, much higher, and you have a better system to make sure that you get DNA in there. Now, would you use a plasmid or would you use a bacteriophage? It really depends upon what laboratory you move into and what their expertise is. Some people like one, some people like the other. Okay, some people do both. But it just depends upon the techniques that your whatever research lab that you go into uses. They're not necessarily better or worse overall, but one might be better for a certain type of work and one might be better for another. All right, those are relatively small pieces of DNA. Okay, sometimes we want larger pieces of DNA, somewhere in the 100 to 300 base pair range. That's a much bigger piece. What we're going to do is we're going to make a bacterial artificial chromosome. Okay, what we're going to do is we're going to start off with a little tiny vector that looks normal, but the way it is constructed, what we can do is insert a very large piece of DNA, and because of what's on that vector, it behaves like it's a full genome. Okay, and so we insert that into the cells. The infection rate is very, very low. Okay, but if you can get it in there, you get one, you can continue to grow, and it can be infective. And so we are now doing a very large piece of DNA that we are copying and cloning. Okay, and that's using bacterial cells. If we want to go even larger, we have to change from bacterial cells to yeast. And again, depending upon what your laboratory works with, we can make a yeast artificial chromosome. We start off again with what looks like a small vector, but it has a couple extra things. Okay, and in particular, what is a CEN? What does that stand for? Centromere. And a TEL? Telomeres. Okay? And so what we can do is we can take this engineered plasmid that starts off with, we can digest it and insert somewhere between 150 and 2 million base pairs. And after it has been processed, it looks just like a chromosome. And that then can be transformed into the yeast cell, and then we can grow up the yeast cell. Here, very large pieces of DNA. And so we can go from very small to very large. Very small tends to isolate one gene, okay? One protein, let's say, that you want to study. The amount of DNA is about the size of a protein. So if I only want to look at one protein, I do that. Okay? And you can look at the original protein, or you can make a mutant of that protein, change the DNA a little bit, and we can do lots of studies. Sometimes, though, we want to take an entire genome and we want to make a copy of that genome and grow lots and lots of it. So we use these other systems like bacterial artificial chromosomes or yeast artificial chromosomes. What we take is the entire DNA and cut it up into big chunks. Then we mix them with the vectors. And what we hope to do is create a genomic library where at least one chunk of every segment gets incorporated into the mixture of vectors. Okay? Genomic libraries are notoriously hard to create, but once you've created them and you have every gene represented in fragments, then they're a wealth of information. Then you can share them with lots and lots of laboratories, and you can get lots of citations because your work is generating their work. And so creating a genomic library is a really important part. And what people have to do is they put the fragments in and they have to then identify, did I have every gene represented? It takes a lot of work to create it and then to prove that you have all the genes there for it to be shared and used. Another functional way that we create cloning vectors is through the creation of cDNAs, okay, or complementary DNAs. Here's the deal. If you are going to work with 1 million pieces of DNA, what you really can't do is this. Go to our genes, our DNA, chunk out a piece, and have a functional protein. Because our genes are not sequential. We have segments that are coded and segments that are not coded and segments that are coded and segments that are not coded. And so we can't just simply go to the DNA, chunk out what I want, and copy it. So what we do is we do take another route. Okay? What we do is we know that our genome is processed through messenger RNA. And in the processing of messenger RNA, those chunks of DNA that aren't expressed are excised. And all the ones that are expressed are condensed into a short piece of messenger RNA. And so now we have a functional piece of DNA. If at that point we could capture that messenger RNA out of the mix and make a copy of it some way, then we would have a short piece of DNA that we can insert into something that is really easy to grow, like bacteria, and we can make a mammalian protein in a bacterial system. If I took our DNA and put it into a bacterial system, it would do the entire chunk of DNA, giving all of the DNA, both the translatable and the non-translatable, all mixed up, and it wouldn't work. All right, and so isolating the messenger RNAs would be a really great method. And then, if I can make a copy of it, well, there are a couple of key points to doing this. All right, how in the world might you extract messenger RNA? What do you know about RNA that might make it extractable out of a test tube? Okay, messenger RNA is copied, right? At the front of the messenger RNA, you learn in genetics, you put a cap on it, right? You've got the sequence, and after it's processed, you got the sequence that will be translatable directly to protein. What's at the end of the messenger RNA? Poly-A tail. Okay? So, your messenger RNAs, every single one of them have a poly-A tail. Well, if I take a poly-T and put it in the mix, that poly-T can be annealed or match up to the poly A. Well, that's not so good if I just put poly T in solution. Now I got things floating in solution. But if I hold on to the poly T in some way, like anchor it to something, and I put the poly T onto there, then the messenger RNAs get trapped onto that something large, and it can be anywhere from a bead to a piece of magnet. And there's really kind of interesting ones where you put poly T on a magnet, you put it in there, and then you use a magnet and you extract everything out. That's kind of cool. And you wash everything away. But you now have lots of messenger RNAs, and that's great. Now, in our genome, in our cells, messenger RNA can only be read really by the ribosome to do its thing and create protein. But I don't want RNA because RNA is not very stable. DNA is what's stable. And so until we found this protein that is used by bacteria, the reverse transcriptase, okay, what the reverse transcriptase can do is take RNA and create a strand of DNA. That's great. DNA is a copy of it, much, much more stable. And then if we process it a little bit more, we take off the RNA or degrade the RNA, we can make a copy of the DNA that you made. And now you have a short piece of DNA that makes your mammalian protein directly. That can be used in bacterial cells or other cells to grow. And so we can make complementary DNAs. Ooh. I have no idea how loud this is, but we're going to try.
A messenger RNA molecule has a 3' poly A tail. So, using an oligo-DT primer and reverse transcriptase, a complementary strand of DNA, or cDNA, is synthesized, forming an mRNA-DNA hybrid. The mRNA strand is degraded with alkali, and the free 3' end of the DNA strand forms a hairpin loop that primes synthesis of a second DNA strand using DNA polymerase III. S1 nuclease cleaves the hairpin loop, leaving a double-stranded cDNA copy of the original mRNA.
Okay, and so now you have a piece of DNA that can be used later. Now, the S1 nuclease, the wrapping around is one technique. There are a lot of different techniques that you can buy from companies. Each one has a kit, and the kit does this. You take your sample, you add in 3 drops of blue, you spin it around, you add 7 drops of yellow, but really it's different enzymes. One of them will be reverse transcriptase in that process. And so one of them will be the poly A tail to isolate them. And so in the book, your figure has the poly T tail going to the poly A, so you've done that. Reverse transcriptase will make the first DNA. They talk about ribonuclease H to degrade, okay, the RNA. And instead of relying on the looping around, which is low efficiency, they are using one of the DNA fragments to serve as the template because to synthesize DNA you need a template or primer. They put in DNA polymerase. It will make your DNA copies. And with the repair mechanism, it'll take out the RNA. Then use DNA ligase to ligate the ends, and at the end you will have your piece of DNA that you want. And so depending upon the kit, the specific enzymes that you use may be a little bit different. Reverse transcriptase is always part of it. The poly T to isolate the messenger RNA is always part of it, but there are other enzymes can be used to do the replication part of the second strand of DNA. And it just depends upon which kit you use. So we can have and isolate proteins from mammalian systems and grow them up in yeast. Or we can grow them up in E. coli, bacterial cells. E. coli is easy to keep alive, cheap to do. We use it all the time. And so that is an easy way to isolate a mammalian protein in a system. It also then makes it easy to isolate because you put a mammalian protein in a bacterial system. There's probably going to be some physical property that the mammalian protein is totally different than the proteins that are in the bacteria. So that allows us to isolate them from. And you're then going to overexpress it. So there's going to be a lot more of that one protein in your bacteria. What is the problem, though, about making your protein in a bacteria, a mammalian protein in a bacteria? We've already isolated the fact of the exons and intron issue that by making the cDNA. Is there any other issue about processing a cDNA of a mammalian protein in a bacterial system?
Not the same machinery?
It's not the same machinery up to a point. Up to a point is perfectly exactly the same. The DNA will be translated, the amino acids will be placed in the right order exactly in both systems. But in the mammalian system, there is what are called post-translational modifications. Things that aren't just the sequence of amino acids, because both will put the right sequence. But in the mammalian system, sometimes those amino acids are made derivatives of, that you have a whole host of other proteins that do that the bacterial system doesn't have. Many times the post-translational modifications don't matter, but sometimes they do. Sometimes that post-translational modification will be on an amino acid that is important to the reaction. And so, yes, it's cheap, yes, it's easy, I can make lots of protein, but you may make a protein that is not necessarily representative of the function of what you need. And so, understanding what your protein actually is for real is important. But a lot of times what we do is we isolate a piece of DNA in the mammalian system that we think we know what's going on, but we don't have a lot of information about it. So we study it in bacteria because it's easier to do, and we do all this work, and then later, we find out, uh-oh, there was something that was a little bit different. And that's where you can get a whole group of labs saying, it works this way, and you get a whole group of labs saying, no, it works this way, and they're totally different. And you have journal articles, both peer-reviewed, both supported by different groups, with two conclusions that are totally different. Not meaning that the science is bad in any one case, but maybe not all of the aspects were taken into account for as you do your very careful work. All right? Okay, sequencing. To actually know the sequence of DNA, that's important because that's where the information is, and we've done lots of sequencing. The largest project that was in terms of sequencing was the Human Genome Project. Again, one of the very few governmental projects that was done under budget and before the timeline that it was anticipated. But we still do lots of sequencing. We actually even do sequencing here. Michael Freak goes out into the rivers with his research students, traps the hellbenders, cuts off their toes, and brings them into the lab, isolates, and he does sequencing downstairs in the 214. We have a DNA sequencer, and so we do it here. Lots of people do it. Okay. In terms of sequencing, here's the problem. It's chemistry-based. Okay. And so when we do something chemistry-based, there's always a limitation. And so in terms of sequencing, what we have to do is we have to make a derivative of the original piece of DNA, and then we have to isolate what that change is down for every base pair. And so in the genome of anything, the genome is so large that we can't just start with one piece and go through the entire genome and sequence it. That's just physically impossible. Okay, so what we have to do is we have to isolate the entire genome. Okay, get it pure. I can't sequence it from the beginning to the end, so if I can't do the whole, what do? I sequence parts. So I have to cut it all up. Uh-oh, cut it up. All right, well, and I get lots of little sequences. And I hope I get it all. But once you cut up the whole piece of DNA and you've got like thousands of linear sequences, now we have a puzzle. What goes what before what? Well, by itself, we have a puzzle that is unsolvable. Okay? And so when you start off with your big piece of genome and you cut it, okay, you have to do that. You have to isolate each cut and you have to sequence each cut. You also have to take that same genome and cut it with at different places. So that what happens is you'll get a fragment that is like this and you'll get a fragment like this, and how do they line up? I have no idea. They can go this way or they can go that way. But if you have another sample that has a piece of DNA that is between these two, I get a sequence of this part and I get a sequence of this part, and if I compare the top one and the two bottom ones, I see, these have to line up this way, because if they go this way, that doesn't match up. And so now it becomes a real big puzzle. You have to sequence it multiple times to get enough information to overlap all of your different pieces that you have. Well, we've got to cut DNA, and we've got real great ways to do that. We use restriction enzymes. We know exactly where they cut, and so we can make and ensure that we have all the pieces. Okay. Once you have a mixture of pieces, you've got to isolate each of those pieces. And to isolate them, we're going to use electrophoresis. Okay? I throw that word in like you know what that is. How many people did an electrophoresis in lab? All right. Which way? Did it lay flat or did it go vertical? You did vertical? Not the flat one? Okay. Most DNA is the flat on the tray, really thick piece of DNA, agarose, and it goes through. Here's the deal. Very quickly in terms of electrophoresis. You got your sample. Okay? You put it in some sort of, it's kind of like Jell-O, whichever one you use. All right? And it's in a solution. So you got a solution full of Jell-O and you put your sample in. Then you plug it up. So that means there's an electrode on one side and an electrode on the other side. All right? And so if I put my DNA here, what electrode do I want on this side? The which one? Positive. Because the DNA is negative because of all the phosphodiesterase. All right, so the positive electrode's here, the negative electrode's there, and the DNA runs towards the positive electrode. Okay, that's great. But if there was not the Jell-O, okay, and the Jell-O can be agarose or polyacrylamide. There are different kinds of Jell-O. All right, that Jell-O matrix have holes in them. And so little tiny things can get through the holes and run fast. Larger fragments run slower. And the screening effect of how large those pores are will get the separation that you need. Okay? And so we're going to use electricity to move things, but the separation will be the matrix by which it moves through. Okay? In terms of how big you want, if you need large fragments of DNA, we usually use what's called agarose. If we want to separate down to individual base pairs, so if I can to separate 9 base pairs versus 10 base pairs, agarose is not small enough to do that. We use polyacrylamide to be able to get to a fine enough grid that I can separate down to single base pairs. For fragments that are less than 1,000 base pairs, you get the polyacrylamide gel. Larger fragments, up to 100,000 base pairs, you can separate them by agarose gels. And then there are real strange things for isolating very large pieces of DNA, including chromosomes. But we usually use agarose or polyacrylamide. Now, here's the deal. If you've ever seen a sample of DNA in a test tube, it looks like you got buffer. Buffer looks like water. Even if you have DNA inside of it, it looks like water. You don't see it. You put it on a gel, you don't see it. It separates, you don't see it. So you have to put something in there to see it. And the most often used, although we don't use it here, is ethidium. Okay? Ethidium cation is mixed into the gel, and we shine UV light on it, and wherever DNA is, the ethidium binds to it and fluoresces. Okay? If it's not bound, it doesn't fluoresce. And so that's a wonderful way to identify where that is. We have alternatives to ethidium bromide. Now, why in the world ethidium bromide actually fluoresces? What happens is it's an organic molecule, it's an aromatic ring, that sticks in between base pairs. When it's stuck in between base pairs, it fluoresces. Okay? And so if I'm trying to find where DNA is, I want to find where the base pairs are, and so it gets in there and gets stuck. We don't use it in this lab, in our labs. Why? Why would it be a bad thing to use in our labs? What does ethidium bromide do? It sticks in where? Between base pairs. Okay? It'll do it in your sample. What if you get it up? What if you get it on yourself? It'll get into your base pairs, and when it's there, it allows derivatives to be made and can be cancer causing. So we don't tend to use it because of how it works. We now use a couple other things that will bind to DNA in a different way, that even if we get it on our hands, it's not going to be cancerous to us, although we don't believe that you should get it on your hands anyway. The ethidium is an older technique. A lot of people use blue safe or other safe, but you go into a research lab, ethidium is probably going to still be there. And they believe, once you're a graduate student, nothing can hurt you because you know what you're doing. We will also use some techniques called southern blotting and northern blotting to actually identify and detect DNA. We're going to transfer in terms of DNA, which is called southern blotting, onto a piece of paper and then analyze it. Northern blotting is if you transfer RNA. There's another blotting technique that you learn. Does anybody know? There's northern, there's southern, and there's what? Western blot. Western does protein, so all the same technique. What's an eastern blot? Something for you to develop. Okay? Why in the world do we use northwest and south as techniques? I mean, you can see that we use a different direction for the three different things. Why in the world do we even go down the north, south, east, and west? Not east, but, you know? No, we don't think there's only 3 ways. It would have been more logical if there were 4. Okay? Dr. Southern developed the DNA. And so from that, RNA was northern, and then when they did it with protein, they just called it western. And so, all right. Here's an overview.
The southern blot is used to verify the presence or absence of a specific nucleotide sequence in the DNA from different sources, and to identify the size of the restriction fragment that contains the sequence. In this procedure, the DNA is isolated from each source and then digested with a specific restriction enzyme. The DNA restriction fragments are then loaded onto an agarose gel, and the fragments separated by electrophoresis according to size, with the smaller fragments migrating faster than larger fragments. The DNA is then transferred from the fragile gel to a nylon filter. Next, the radioactively labeled nucleic acid probe is added. The probe binds to complementary DNA segments. Note that the DNA segment being probed is not present in organism B. To detect the position of the radioactive probe, the nylon membrane is covered with an X-ray film. After development, the positions of the probe become visible.
Okay, I think that's it. All right. In this case, what you're doing is you're separating your sample. You hope that on that sample there's a piece or target piece of DNA that you're looking for. Okay? You need to know what you're looking for, because what you're going to do is you're going to order a sequence of DNA that's a complement to that. Now that sequence of DNA in this example is radio labeled. A lot of laboratories still use radioactivity. But quite a number of people have moved away from that. They will do a fluorescence tag or a chemiluminescence, where I can either have fluorescence, I shine a light on it and it shines a different wavelength, or I put in a solution and it'll create light. I can still use X-ray film to expose the light, or you can use an instrument to detect the fluorescence. Doesn't matter if it's radioactivity or other technique, just depends upon what you have used. What's important is you separated the pieces in the acrylamide, okay, or the agarose. Now, the problem is you've got inside that gel, that jello, your pieces. You've got to transfer it to a piece of paper. And what they did is they laid that piece of paper on there and they said, oh, it automatically happens. No, what you do is you put a bunch of paper towels. Then you put your little piece of nitrocellulose paper on there. Then you put your gel on top of that. Then you put a piece of paper on top of that. Then you put lots and lots of paper towels on that. And then you take your biggest book you have and you put it on top of it. What happens is you squeeze out all the material and it flows through the nitrocellulose paper and then it adheres to the nitrocellulose film. You get a portion of each piece of the DNA. Then you take all that apart. Now you've got a piece of paper. Piece of paper is easy to manipulate. You put it in a tray in solution and you can wash it and dump that out, and then you can put it into a solution with your probe in there and let it mix, and then you can pour that out and you can wash it, and the only radioactivity or the only signaling molecule will stick if and only if it's actually making a complement, not just adhering. Before you can actually mix the probe, remember DNA is double helixed, so what you have to do is you have to make it single. So before you actually add the probe, you add in a little alkali to make sure that it unravels, okay, so that it's available and open so that your probe can get in there. Once you have that probe and it binds, then the X-ray film or the detector that you put it into in terms of an instrument will identify where on that sheet was the DNA, and you can say that the sample that you have does have your DNA in it, and hopefully you didn't use your entire sample because now you totally destroyed it. And so, but if you go back to your original sample, I have my sample, it's got the thing I want, okay, I can isolate it from that point in time. All right, making copies or making a sequence. There have been lots and lots of techniques. The Sanger dideoxy method is probably the most standard technique that is used now. We used to teach a different method that is so old that very few labs use that technique. The Sanger dideoxy has been automated in lots of ways and is used in most sequencing events. What we're going to do is we're going to take the original piece of DNA that we want to sequence and we're going to make copies of it. Okay? And we're going to make copies in a way that we can detect where each different base is incorporated. To do this, what we're going to need is a sample of the DNA that you want to identify. We need some sort of primer so that we can start sequencing. We need all the 4-deoxy nucleosides so that we can make copies, so we need all the base pairs. Then we need a tiny, tiny amount of what is called 2', 3' dideoxy analog, or the dideoxy method. All right. If I have two oxygens here, I have ribose. If I take off an oxygen or hydroxide here, I've got deoxyribose. And if I take off both hydroxides, I've got dideoxy. Okay. This has a consequence. As I'm adding base pairs, if I have a 3', I can add another one. If I have a 3', I can add another one. But if this one gets incorporated, I have a 3' proton. I stop the sequencing. Even if I have all the other pieces, I have an enzyme to do the sequencing. I have all the nucleotides. I have a sequence and a primer. Once this is incorporated, I've stopped. The other item that you need in this is DNA polymerase to do the sequencing. And so what we have is, we have the sequence that we want, a little primer, the DNA polymerase, all the dideoxys, and then a dideoxy analog. And what will happen is, I will allow the reaction to occur at a certain temperature that optimizes the polymerization. And so it'll go down and it'll add a G where a C, it'll add a C where a G, and it'll go, and then it will add this dideoxy randomly. The randomness is based upon how concentrated you put this in. If this is way too concentrated, you get really, really short segments because you're always incorporating it. If you don't put enough in here, you won't hit every time A is represented in the DNA. So there's a optimal concentration. Once this is stuck, this is done. What you have to do is you have to heat up the mixture, allow everything to melt, come apart. You cool it down so that it anneals the primer back, and then you do some more synthesis. And you make copy, you make copies, you make copies, you make copies. If the concentrations are right, you will have a dideoxy incorporated every time that it's going to be represented in the piece. And you'll have lots and lots of different copies. All right? And you make a bunch of this. It takes a little bit of time. Now, once you have lots and lots of sequences, they're all different sized. Okay? But if you do separation, you should have one that has 4 and one that has 5 and one that has 6 and one that has 7, and you'll continue to make DNAs, one represented for each size. If you start doing separation, you'll separate them by size, and we're going to identify where those dideoxy methods, the dideoxy is actually going to show. There are 2 different ways that we're going to do this, and I will show you those next time. And since I